Hello, good morning to all. Myself, Dr. Charan Kamal Sekhon. I have come all the way from India, maybe most of you must be knowing, and I'm here to present my presentation, and my topic is an inventory of family Noctuidae from Himachal Pradesh. So let's begin. This is the name of my university. And this is the map of India and Punjab highlighted, the state, the province I'm coming from. And from Punjab, there is a city, Fatehgarh Sahib, where I work. Fatehgarh means a town of victory. It has a long story related to Sikh religion, so it's named after that. This is my university campus where I work, and this is the campus view. So this university is named after Sikh holy book, Guru Granth Sahib Ji. It was placed in Golden Temple, hope you'll be knowing it, Golden Temple in 1604. So this is the second university in the world named after a holy book. Yeah. So we are here to discuss the things about biodiversity. So while talking about biodiversity, what I feel is the variability among living organisms from all sources, means including terrestrial, aquatic, and all living forms existing here, between the species and between the groups as well. So why do we gathered here for the biodiversity? What is the concern over it? So the concern is it's a vital resource for all mankind. We need it, right? So its importance leads to its overexploitation. So more we need, the more we have exploited it for our users. So there's a limit to every disruption, and probably we have reached somewhere to that level. So why taxonomic studies? I am a taxonomist. I work in the field of taxonomy, and how taxonomy is connected with biodiversity. Why I'm doing taxonomy is it's a necessary for the estimation of biodiversity. So we need to assess the biodiversity for its conservation, and how we can estimate that for the further planning strategies is, so taxonomic studies include survey and collection, then is monitoring, then followed by mapping. Then once we are done with the mapping, we'll go for a conservation of habitat, then in a habitat conservation of a host plant, which finally going to lead to conservation of a specific species we are concerned with. So starting with a little bit of introduction. So insect, they are very highly specialized group, especially the arthropoda, because they are highly species specific, right? Lepidoptera comprising moths and butterflies, and I have been working since 2005 on moths of family Noctuidae. So why family Noctuidae is? It's because it's a highly economically important group, and they are basically known as owlet moths because their eyes more or less resembles like owlets, right? So their numerical strength has been estimated to be around 35,000 described species, and still many more are awaiting discovery. So during my work, I have studied more or less like 150 species in the tenure of 5 to 6 years till now. But over here I have compiled my work concentrating on one state, that's a Himachal Pradesh only. So why I'm taking this group is because it's a highly economically important group. Most of the pest species belong to family Noctuidae, like we see some of them. Agratus flammetra, very common name. It's a pest of pea and a gram. Agratus epsilon, pest of potato. Then Sesamia and Achia, pest of rose and pomegranate. So these are the few examples, and a commonly found pest in Punjab as well as in Himachal Pradesh and surrounding areas. So I was discussing about the taxonomy procedure, how I've done my work. So firstly, how we do it. The methodology comprises collection of noctuid moths, and then their stretching procedure for their preservation, proper curation, and then is there little bit of layman sorting, and then final identification in the lab from the old literature and the recent literature research, then finally comparisons in national museums and international museums. So various survey collection tours were conducted in the breeding seasons of Himachal Pradesh, and almost like 2,000 or 3,000 specimens were collected with the field collections. So the taxonomic treatment includes collection, killing, stretching, preserving, and then sorting. So this is how I used to collect moths on a vertical white sheet at a night time, as they are very much attracted to moonlight, so they get confused, and we collect them at the night around 7 or 8 o'clock in the evening till 2 or 3 in the morning, and then the procedure comes off like this. Is a taxonomic treatment that we stretch their wings and other body parts so that it can be easy to study afterwards till million of years. So the stretching procedure is like this. This is how I used to spread them, pinning, and I used to keep it for like 24 hours, and once they get theirs, like everything got placed, and then the pins used to be out, and the specimen is, sorry, I have a bad throat. So it was kept in the museum for the further studies. So the taxonomic characters which I used to take for the study, which are species specific, and they are solid characters, right? So these include morphological characters. Basically, the first identification procedure I followed is color patterns on their wings, and then the labial palpi, legs. Thank you so much, sir. Antennae, wing venation, that carries very important role, and most of that, the most important character I have worked on is external genitalia, which I told you earlier as well, that's the external genitalia characters are highly species specific. I have done almost a number of genitalia dissections, and I published many new papers on a new species because externally they were similar, but I found many changes in their external genitalia. So this is this is the classification I followed. So in a field, how do you identify it's a noctuid moth? They are usually dull and drab color, very boring kind of moths, and their sitting pattern is like this. Like while they sit, they fold their wings above their abdomen and making a triangle shape. So this was a study of wing venation. And there's one important principal character, that they have a presence of tympanum on the metathorax. But usually we never go for this character because externally it's very easy to make out from their eyes that it's a noctuid moth. So identification was done with the relevant literature by Hampson and recent literature, and then all the identified species were taken to Zoological Survey of India and Indian Agriculture Research Institute, New Delhi, and Forest Research Institute, Dehradun, within India. And then the collection was taken to Natural History Museum, London, by my colleague for the final authentication and for the final identification and comparison whether we have done a right job or not somewhere. So this is the dissected male genitalia of a noctuid moth. So earlier we used to do the ink diagrams. Now we have a good instrument to get it photographed live immediately after we dissect it. So this is a male genitalia, and these are the important parts which really makes out difference. So that UN is a uncus, male organ part, to hold to female genitalia. The shapes of uncus actually really works well, as it has got a great variation in a same genus but in different species. To differentiate closely allied species, this part carries a highly important. So as the structure of VLV, that is valve, and SA, that is Sachus. I'll show you up. So this is Uncus. Just you can make out the difference shapes in the Uncus in a very closely allied species. So there's a high variability among them too, and it really makes you easy to differentiate, and it's highly authenticate. So this is the corpus bursae, the lower portion of a female genitalia, and the daughter portion you are saying on it is a signum, and the shape of signum, presence of it too helps to differentiate two different closely allied female species. So this is how earlier I worked for a year on an internal genitalia, but I couldn't find it that it has not presented a strong evidence to differentiate. Then I switched over to external genitalia. So this is the procedure to dissect out, given by Robinson in 1976. Then I came to a conclusion of an inventory of family Noctuidae belonging to superfamily Noctuidae from Himachal Pradesh. And 69 genera have been studied during the survey, and 10 of them found as a new record from that area. So this is a list of 105 species collected, recorded, identified, and compared. So the new records are labeled over here in the list. These were not available in that or in nearby surrounding areas. Probably they were present at another corner of the country, but they have not been reported in like many years. So to authenticate my inventory, I have done the external genitalia of all 105 species. So here I'm presenting just one or two slides, not to make you much bore. So all the three species belong to same genera, that is Mithmina, with the different species, and you can make out the different genitalic structures in all the three species. The male genitalia is more or less similar with a little bit of difference in the valve, but in the third species altogether different. So exactly same in the case of genus Leucaena. So here you can find out a great difference between the structures of male genitalia right over there. But they have got some more or less similarity in the female genitalia, but there's a great difference in a male genitalia. So conclusion is, it's a high time to reset our priorities and make a beginning to give due importance to research in field of taxonomy. We have lost so many million of organisms like this, and we cannot afford to lose many more. So we should act now to regain the past glory of systematic studies in order to save not only our biodiversity, but also ourselves. Thank you so much. In last words, taxonomy is not the only science on which castles of other sciences can be built. These are the references I have taken. Thank you so much for kind hearing.
Thanks a lot. Give a big hand again to Dr. Sekhon. We have time now for questions.
Yes, I appreciate your talk because you proudly tell you are a taxonomist. Because nowadays the people are thought that the molecular taxonomy is the one they are following, and if you are doing in the molecular, then only you will get a very good number of paper in it.
I know.
But you are restricted to the this system in the morphological system, and you recorded some new species. I appreciate your thing. And in your view, how you are correlating the traditional taxonomist and the molecular taxonomist, which is the one is the better one? Yeah.
Yes, thank you so much for your question. So that's what I have concluded in my last words, that taxonomy, what I feel, is a tax on me. For me, taxonomy is a tax on me. If taxonomy is a science which actually without taxonomy we cannot build castles, right? So it's a basic science. We cannot begin from just a molecular level to above levels. We have to go from the bottoms, and the things, the people, the study which grows up from the bottom, I guess that has got a strong base, and you can conclude something with a solid evidence. So it's a basic science. I don't feel somebody who's working over molecular, they should basically concentrate on a taxonomy first before going for a molecular study. Merely taking any species and working over their genes just by knowing that it is called as this species, and I don't feel, I personally don't feel it's honest to work on something you are not sure about.
Thank you very much. In fact, I want to say that this is a very impressive biodiversity conservation work.
Thank you so much.
Thank you for the efforts you have put in.
Thank you, sir.
Because the basic ingredient of conservation is to know initially what you are trying to conserve.
Yes, sir.
And that you have been able to discover species that have not been recorded in the environment before is an achievement.
Thank
Just
you
keep
so
on
much.
the work.
Thank you so much. That's what most of my colleagues working. I'm not blaming somebody who's working on molecular biology. They are good people. Rather, they must be more intelligent than me that they are working on a molecular, because I hate that genes work. So I appreciate their work as well. But what I think is taxonomy, it gives you a solid evidence, right? It's very important. We should not leave our roots. It's a basic root to science. So to begin with anything, any work.
Dr. Sekhon is one of the premier in your area.
Oh,
Nice
to meet you, sir. Yeah, sure, sir.
And he doesn't do research really in the lab.
Okay.
He knows how to apply that in the field.
That's what I was supposed to say, sir, when I used to collect them, and most of my colleagues say how much importance does it carry, the authentic work, the new work, and the implementation relies on molecular biology or biochemistry. But the authentic identification is must to begin with something. Yeah, I know I don't listen, man. That's the only reason. I mean, you really need authentic identification for the conservation, and that too implementation.
Yeah, we have time for questions.
Yeah.
Okay, yes.
Thank you. So you wanted to be labeled as a Noctuidae taxonomist?
Yes, sir, I proudly say that.
Okay, one thing. And your work is totally confined to nocturnal moths, or
Yes,
including diurnal moths also?
No, sir, noctid moths, basically.
Only nocturnal.
Actually, this is a large group, and it was almost messed up. It took me 2 to 3 years just to collect the old literature over it. It comprises 1912 species within India.
As far as India is concerned, there is very little work done on this.
Yes, sir, very little work. I mean, actually, this is a messed up family, so people really don't like working on it. But being a Punjabi, I belong to Punjab, being related
First
to
Indian, then Punjabi.
Yeah. So what I faced is our farmers, they are really suffering from these pests.
Yeah.
So yeah, earlier I was interested to work on mosquitoes, then I switched over to my mind.
And one other point. So you said that you have collected specimens.
Yes, sir.
And took them to IARI.
Yes, sir.
And then ZSI.
ZSI.
And then to London.
No, sir, my colleague
Your
went
colleague.
to NHM.
So, and that is one side.
Yes.
The other side, you say you used all these genital characteristics for the identification.
Yes, sir.
So you say that you have identified using these characteristics, and again, what is the need for you to take them to these?
Actually, I was doing my doctorate at that time, and there's a stage till you don't feel that confidence level until you don't conclude everything. So meanwhile, my colleague went to, I done my all the identification on wing venation, on the base of wing venation, right? So I was doing my genitalia part. Meanwhile, she went, we both were planned to went to London, but I couldn't go for some reasons.
Yeah, okay, that's
So
good.
she took my collection.
Did you get permission from the Biodiversity Authority of India to take specimens?
Yes, sir. It was a difficult task. We had a conversation with the.
We will wrap up. We will have a discussion later between you and.
Yeah, but before we go, I want to allow him to understand there is always room for confirmation.
Yes.
Yes.
Especially in taxonomic work, you do your identification, you need a second, a third, and a fourth
Yes,
opinion
that is the reason
before
she
you
did
can.
that,
So
but
what
if-
you are doing is perfect.
I know.
If you
Thanks
have
so much.
argumentative questions, we can do that later, not in the forum here. Outside, we can always back and forth, we can talk. No. We cannot ask like too long questions. Just go for direct questions and get answer.
Thank
Thank you.
you
Thank you
so
very
much
much.
for your kind hearing, lovely people.
I just
It's
want
really
to-
nice to meet you all, and I'm really pleased to hear, and I really make sure that I see you all again on the fifth conference. Thank you
Dr.
so
Sekhon.
much.